Welcome back to the Deep Dive. Today, we're shifting gears a bit. We spend so much time on this show talking about the microscopic engines of life, DNA, mitochondria, protein synthesis, the really small stuff.
We do.
But today, we are zooming out. We're going to look at the logistics of survival.
It's a necessary shift. I mean, you can have the most perfect, pristine cells in the world, DNA intact, organelles just humming along. But if the supply chain fails, it's all over.
Right. It's the last mile problem of biology. Today, we are talking about the plumbing, the pressure, and, well, the blockages. We're covering hemodynamic disorders, thromboembolic disease, and shock.
And this is the big stuff.
It really is. Frankly, looking at the data, this is the stuff that actually kills people in the modern world. Myocardial infarction, pulmonary embolism, stroke, these aren't cellular defects to start with, they are flow problems.
Exactly. The circulatory system is a high-pressure, closed-loop hydraulic system, and it relies on this very, very precarious balance of physics and chemistry.
Yeah.
When that balance tips even slightly, you don't just get a local issue, you get systemic collapse.
To guide us through this, we are synthesizing two of the absolute heavyweights in pathology. We have the Bible, Robbins, and Cochrane pathologic basis of disease.
Of course.
Which gives us that incredible systems level view, the clinical integration.
And we're pairing it with Underwood's pathology. I love Underwood because it's so grounded in mechanism. It constantly reminds us that disease is just a physiological response to injury.
It frames everything
as the body's trying to fix X, and in doing so, it accidentally causes Y. It's all cause and effect.
Before we dive in, the standard disclaimer.
Right.
We are building a conceptual map here. This is a synthesis to help you revise and understand the why behind these conditions.
Right.
It is absolutely not a substitute for sitting down with Robbins or Underwood and actually reading the chapters on hemodynamics.
Yeah.
Consider this the scaffolding.
You still have to lay the bricks.
Exactly. So we have a massive itinerary today. We're going to start with the physics of fluids, why water stays in your veins, and what happens when it leaks out, what we call edema.
Then we're moving on to the solid state, thrombosis. Why liquid blood turns into a rock.
And we'll break down Virchow's triad there, which is just, it's essential.
Can't talk about thrombosis without it.
Then we're going to track that rock as it travels, embolism. And finally, we're going to spend a significant amount of time on the end game, shock. And I don't want to just list the types of shock.
No.
I want to understand the cellular suicide that happens when perfusion fails.
That's the most critical part. Shock isn't just low blood pressure, it's metabolic death at the cellular level.
Okay, let's start with the basics. Part one, the physics of fluids. I was reading the section in Robbins on edema, and it struck me how counterintuitive the human body is. We're mostly water, right?
Right.
And we have liters of blood pumping at high pressure through these really thin-walled vessels, yet our tissues are normally dry, or at least they aren't soggy.
It is a bit of a magic trick, isn't it? It all comes down to something called Starling forces. It's a tug of war happening in every single capillary in your body, every second of every day.
A tug of war. I like that. So let's unpack the players in this tug of war. On one side of the rope, we have hydrostatic pressure.
Right. Think of hydrostatic pressure as the push. Your heart is a pump. It drives blood into the arteries and down to the capillaries. That physical pressure, just the force of the fluid, wants to push water out of the vessel and into the surrounding tissue.
It's like a garden hose with tiny pinpricks in it.
That's a perfect analogy. If you crank up the tap, water is going to spray out. That's hydrostatic pressure.
Okay, so if that were the only force, we'd all just swell up and, well, pop. So there has to be an opposing force, a pull force keeping the water in.
And that's the plasma colloid osmotic pressure. Sometimes you'll hear it called oncotic pressure. This isn't physics so much as it is pure chemistry. It's driven by the proteins dissolved in your blood plasma.
And one protein in particular does most of the work.
One protein does about 80% of the heavy lifting, albumin.
Albumin is fascinating. It's basically a molecular sponge floating in your blood.
That's the perfect way to think about it. Albumin is a big molecule. It sits inside the blood vessel, and it can't really fit through the pores to get out into the tissue.
It's trapped.
It's trapped. And because it's trapped there, it draws water towards it through osmosis. So you've got this beautiful balance. Hydrostatic pressure pushes water out. Albumin pulls water back in.
And in a healthy person, these two forces almost cancel each other out.
Almost. And that almost is key. There is always a slight net loss of fluid out into the tissue. The push is always just a tiny bit stronger than the pull.
Which brings us to the third player in this system, the real unsung hero, the lymphatics.
The scavenger system.
Right.
The lymphatic vessels are these tiny little channels that are constantly vacuuming up that extra bit of leaked fluid and, crucially, any stray proteins that might have snuck out. And they pump it all back up to the thoracic duct and return it to the venous blood. It's the safety valve.
Robbins makes a point that I think gets overlooked sometimes. The interstitium, that space between the cells, it's not just empty space, it's a gel.
Yes, the interstitial matrix. It's essentially a hydration gel made of proteoglycans. This is important because it provides some initial resistance. It limits how much fluid can accumulate at first.
But it has a limit.
It has a limit. Once you overwhelm the lymphatics and you fully saturate that gel, you get frank, pitting edema. That's when you can press your thumb into someone's shin and leave an indentation.
So edema is just the failure of one of these mechanisms. It's not a disease in itself. It's a sign that the physics is broken.
A very important clinical sign.
So let's look at the clinical scenarios, because this is where the detective work happens. Let's take the classic example, heart failure.
Classic. The pump is broken.
So in heart failure, the heart can't pump blood forward effectively. So naturally, blood backs up in the veins. That backup increases the hydrostatic pressure.
The push gets too strong.
Right. The pressure in the capillaries rises, and fluid is forced out into the tissues. Legs swell, lungs can fill up with fluid. But Robbins points out that it's not just the back pressure. The kidneys actually make it worse.
This is the real tragedy of physiology. We call it forward failure. When the heart fails, it doesn't pump enough blood forward to the rest of the body. The kidneys, which are constantly monitoring blood flow, interpret this as, oh my God, we are bleeding to death. We are dehydrated.
They think the tank is empty.
But the tank is full. The pump is just broken.
But what do the kidneys do?
Exactly what they're programmed to do in that situation. They activate the renin-angiotensin-aldosterone system, the RAAS. The juxtaglomerular cells sense low flow. They release renin. And the end result of this whole cascade is that the kidneys aggressively hold on to salt and water.
Which increases blood volume.
Right. In a hemorrhage, that would save your life. It would refill the tank. But in heart failure, you are dumping more fluid into a system that is already backed up and overflowing.
You're just making the hydrostatic pressure even higher.
It's a vicious maladaptive cycle. The body's own compensation mechanism ends up drowning the tissues.
And that's why we give patients diuretics and ACE inhibitors to block that very response from the kidney to stop it from helping. Okay, now let's flip the equation. What if the push is fine, but the pull fails? What if we lose the albumin?
Then you have no sponge. Water just leaves the vessel because there's nothing holding it back. Osmotically, there's no reason for it to stay. This happens in two main ways.
Okay.
Either you aren't making enough albumin in the first place, which points to severe liver failure, like cirrhosis.
Because the liver makes albumin.
Correct. Or you're making it just fine, but you're peeing it all out. That's nephrotic syndrome, a kidney disease where you lose massive amounts of protein in the urine.
And the clinical presentation is different, isn't it? Heart failure edema is usually gravity dependent. You see it in the legs and ankles.
Correct, because the pressure is highest down there. But if you lose albumin, the pull is gone everywhere. The oncotic pressure is low system-wide.
So you get generalized edema.
You see it in the face, especially around the eyes, periorbital edema. You see it in the hands. You get ascites, which is fluid in the abdominal cavity. It's a total system failure of fluid retention.
Before we leave fluids, we need to clarify red versus flu. The texts distinguish between hyperemia and congestion. It sounds simple, but the mechanism matters.
Oh, absolutely. And please, let's go deeper than just the color.
Agreed. So hyperemia is active. It's me blushing because I'm embarrassed, or my muscles getting engorged with blood when I'm lifting weights.
It's an active process. It's arterial dilation bringing fresh, oxygenated blood in to meet increased demand. It's physiological.
And the tissue turns red because it's full of oxyhemoglobin.
Right. Congestion, on the other hand, that's the nasty one. It's passive.
It's a clawing backup.
Exactly. The exit is blocked. Usually it's a venous obstruction or, again, heart failure. Deoxygenated blue blood piles up because it can't get out.
So the tissue turns bluish or cyanotic because the hemoglobin has given up its oxygen and is just sitting there stagnant.
But the histology,
Yeah.
the changes you see under the microscope, that's what really tells the story of chronic congestion.
This leads us to the classic nutmeg liver. I feel like this is a rite of passage for every medical student identifying this in a jar.
It is. It's the classic macroscopic hallmark of chronic passive congestion of the liver, almost always from right-sided heart failure.
So describe what we're looking at mechanically. Walk us through it.
Okay, so the right side of the heart fails to pump blood to the lungs. Blood backs up into the big veins, the vena cava. From there, it backs up into the hepatic vein and then into the tiny central veins at the center of each liver lobule.
So the very center of the functional unit of the liver gets backed up.
It gets engorged with deoxygenated blood. The pressure rises, and the liver cells, the hepatocytes, right around that central vein start to die from hypoxia. That's called central lobular necrosis.
And those dead areas are the dark red or brown spots.
Yes. But the hepatocytes on the periphery of the lobule, the ones closer to the arterial supply from the hepatic artery, they survive longer. They might just get stressed and undergo fatty change, which makes them look pale.
So you get this mottled pattern.
A pattern of dark, dead, congested centers surrounded by pale, fatty halos. It looks just like the cut surface of a nutmeg, hence the name.
And if you look under a microscope at the lungs in chronic congestion, say from left heart failure this time, you see something called heart failure cells.
Another classic board exam question. It's the same principle. The left heart fails, blood backs up into the pulmonary circulation. The capillaries in the lung get congested and pressurized for so long that they eventually rupture.
They start leaking.
They leak red blood cells into the air sacs, into the alveoli. The body's cleanup crew, the alveolar macrophages, come in and they eat those escaped red blood cells.
And red blood cells are basically little bags of iron.
Exactly, in the form of hemoglobin. So the macrophages get stuffed with this rusty brown iron pigment called hemosiderin. When you look at a lung slide, you see these brown granular cells all over the place. Hemosiderin-laden macrophages.
It's the histological proof.
It's forensic evidence that the patient's heart has been failing for a long, long time.
It's amazing how the body leaves these clues behind. Okay, let's move from fluid dynamics to solid-state physics. Part 2. Thrombosis.
The clotting system.
Underwood describes hemostasis as existing on a knife edge. And that image really stuck with me. Blood has to be a perfect liquid to flow through miles of capillaries.
But the second there is a hole, it has to turn into solid concrete. And it has to do it only at the site of injury.
It's all about localization.
If it clots everywhere, you die. If it clots nowhere, you bleed to death. The regulation has to be incredibly tight.
First, let's make a key distinction. A clot versus a thrombus. Before reading this chapter again, I have to admit I use them interchangeably.
Most people do, but in pathology, they mean very different things. A clot is just coagulated blood. It can happen in a test tube, or it can happen in your body after you die, a post-mortem clot.
And what does it look like?
It's gelatinous, it's unstructured, it often has a yellow chicken fat supernatant, and crucially, it is not attached to the vessel wall, it's just a cast of the vessel.
Whereas a thrombus?
A thrombus is a completely different beast. It is a pathological structure formed in leaving flowing blood, and because the blood is constantly flowing past it as it forms, it gets built in layers.
This is where we see the lines of Zahn.
Exactly. If you slice open a thrombus and look at it, you see these distinct alternating layers. You see pale layers made of platelets and fibrin, and then dark red layers of trapped red blood cells. Pale dark, pale dark. It looks like sedimentary rock.
And that's the telltale sign for a pathologist.
It's the absolute proof. If I'm doing an autopsy and I find a mass in the pulmonary artery and it has lines of Zahn, I know for a fact that it's a pre-mortem thrombus that embolized and killed the person.
It looks like jello without layers.
Then it's just a post-mortem clot that formed as the blood settled after death, not the cause of death.
So what causes a thrombus to form in a living person? We have to bow down to the master here. Rudolf Virchow.
The father of modern pathology. A fascinating character, actually. He was also a progressive politician in Berlin, fought for public health reforms, but his lasting legacy in this field is, without a doubt, Virchow's triad.
The three corners of the triangle. If you have these conditions, you get thrombosis. Corner 1, endothelial injury.
The most important one, by far. The endothelium, the single layer of cells lining all your blood vessels, is not just a passive wrapper. It is a highly active chemical factory.
Under normal conditions, it's anticoagulant.
It's actively anti-clotting. It expresses molecules like thrombomodulin. It produces nitric oxide and prostacyclin. It actively prevents platelets from sticking and clots from forming. It's like a perfect non-stick Teflon pan.
But if you scratch the Teflon...
If you damage it through the sheer stress of hypertension, toxins from smoking, high cholesterol causing atherosclerosis, or just physical trauma, you expose the subendothelial collagen. That's like a bug light for platelets. They stick immediately.
But Robbins points out something deeper. Endothelial activation. The cells don't even have to be physically ripped off.
This is a key concept. If the endothelial cells get inflamed, say from cytokines or bacterial toxins, they change their gene expression. They downregulate the anti-clot stuff and start upregulating pro-clot stuff like tissue factor.
So an angry endothelium is a prothrombotic endothelium.
Precisely.
Okay, corner 2. Abnormal blood flow.
Stasis or turbulence. Blood is meant to flow in a smooth, streamlined fashion. We call it laminar flow. The cells are in the middle of the stream, and the plasma is on the edges, insulating them from the wall.
Stasis is the big enemy in the veins, right?
It is. Think about the deep veins in your legs. They don't have a big pump like the heart pushing them along. They rely on your calf muscles squeezing them every time you take a step.
The calf muscle pump.
Right. So if you sit on a 14-hour flight to Australia, or if you are bedbound in the hospital after a major surgery, that pump stops working. The blood pools. It becomes stagnant.
And what does stasis do?
2 things. First, platelets, which are normally in the center of flow, drift out to the edges and make contact with the endothelium. Second, activated clotting factors, which are normally washed away by flow, start to accumulate to a critical concentration.
And that's the perfect recipe for a DVT, a deep vein thrombosis.
Exactly. Turbulence, on the other hand, is more of an arterial problem. If you have an aneurysm, a ballooning of an artery, or a jagged atherosclerotic plaque, the blood flow becomes chaotic. It swirls.
Like eddies in a river.
Perfect analogy. Those eddies can directly cause endothelial injury, which is corner 1, and they also create local pockets of stasis within the turbulent stream. So turbulence promotes thrombosis through 2 different mechanisms.
Okay, that makes sense. Corner 3, hypercoagulability. The blood chemistry itself is just wrong. It's too sticky.
This is where we get into primary and secondary causes. Let's talk about the most common genetic one because it's so important. Factor V Leiden.
This is a mutation in one of the clotting factors, factor V.
Right. So normally your clotting cascade has checks and balances. It has off switches. One of the most important off switches is a molecule called protein C. The job of protein C is to find activated factor V and chop it up, inactivating it to stop the clot from growing too big.
So protein C is the brakes.
It's the brakes. But in people with the factor V Leiden mutation, the factor V molecule is shaped just slightly differently. It has a single amino acid substitution, and because of that change, it resists being cut by protein C.
The brakes don't work.
The brakes fail. The clotting cascade keeps firing long after it should have stopped. It's a lifelong prothrombotic state.
And on the acquired side, we have to talk about HIT, heparin-induced thrombocytopenia. This is one of those medical paradoxes that just blows my mind every time I think about it.
It is genuinely terrifying. So you give a patient heparin, which is a powerful blood thinner, usually to prevent clots after surgery or for a heart attack.
You're trying to do the right thing.
Exactly. But in about 5% of patients, their immune system freaks out and develops an antibody. This antibody doesn't target heparin itself. It targets the complex formed by heparin and a protein released from platelets called platelet factor 4.
And usually antibodies neutralize things. They get rid of them. But this antibody...
This antibody is different. When it binds to the heparin PF4 complex on the surface of platelets, it acts like an activation switch. It turns them on, all of them.
So you get massive system-wide clotting.
Catastrophic arterial and venous thrombosis while the patient is on a blood thinner.
And the name includes thrombocytopenia, which means low platelets. Why is that?
Because all the platelets are being consumed in these millions of tiny clots. So when you check their blood count, their platelet count has plummeted. You have a patient with a low platelet count who is simultaneously clotting to death.
A nightmare scenario.
It requires immediate recognition, stopping the heparin, and switching to a completely different class of anticoagulants. It's a true medical emergency.
Okay, so Virchow's triad has done its work. We have a thrombus, a solid rock in a liquid stream. What happens to it next? What's its fate?
The thrombus has an itinerary. Robbins lists 4 main fates. The best-case scenario is dissolution. The body's own fibrinolytic system, using an enzyme called plasmin, successfully dissolves the clot. Everything goes back to normal.
That's what we hope for.
Always. The second fate is propagation. The clot continues to grow. It accumulates more platelets and fibrin, often trailing like a long tail in the direction of blood flow towards the heart.
Getting bigger and more dangerous.
Right. Third is organization and recanalization. The body basically accepts that the clot is there to stay and treats it like an injury. Inflammatory cells grow into it, turning it into a scar that becomes part of the vessel wall.
And recanalization.
Over time, new, smaller capillary-like channels can drill their way through that scar tissue, trying to restore some blood flow. It's never as good as the original, but it's an attempt.
And the fourth, and often worst, fate,
Embolization.
the traveling thread. This brings us to part 3.
An embolus is simply an intravascular mass solid, liquid, or gas that gets carried by the blood from its point of origin to a distant site.
And most of the time, it's a piece of a thrombus.
99% of the time, it's a thromboembolus, a thrombus that has packed its bags. It breaks free from the vessel wall, and the physics here is dead simple. It floats downstream through larger and larger vessels until it inevitably hits a vessel that is smaller in diameter than it is.
And then impact.
Blockage. Occlusion.
And where it lands depends entirely on where it started. So let's trace the path of the most common lethal one, the pulmonary embolism, or PE.
Okay. So we start in the leg. A DVT in the deep femoral vein, let's say. A piece of that thrombus breaks off. It's now an embolus.
And it starts its journey.
It travels up the femoral vein, into the iliac vein, and then into the huge inferior vena cava. The vessel gets wider and wider so it floats easily. It enters the right atrium of the heart,
Passes through the tricuspid valve.
into the right ventricle, still plenty of room. The right ventricle contracts and pumps it out into the main pulmonary artery.
And that's where the road starts to narrow.
That's where the trouble begins. The pulmonary artery tree branches, and the vessels get progressively smaller. Sooner or later, that clot is going to get stuck.
Now, Robbins talks about the saddle embolus. It sounds kind of Western, but it's actually a catastrophic event.
It is the absolute worst-case scenario for a PE. A saddle embolus is a clot so huge that it doesn't get stuck in one of the smaller branches. It gets stuck right at the bifurcation where the main pulmonary artery splits into the left and right pulmonary arteries.
So it sits across the split like a saddle on a horse?
Exactly. And in doing so, it blocks blood flow to both lungs instantly.
This isn't just shortness of breath anymore.
This is acute right heart failure, or acute cor pulmonale. The right ventricle is trying to pump against a completely blocked wall. It can't. It dilates and fails. No blood goes to the lungs to get oxygenated.
And therefore, no oxygenated blood returns to the left side of the heart to be pumped to the brain and the rest of the body.
The result is something called electromechanical dissociation. You can see the heart's electrical activity on an EKG, but it's not actually moving any fluid. It's beating in an empty chamber, essentially.
The patient collapses.
And dies within seconds to minutes. It's one of the most common causes of sudden death in people who might have seemed otherwise healthy, say, after a long plane ride or a few days after a hip replacement surgery.
And now, as you said, 99% of emboli are blood clots, but that last 1%, the exotic emboli, those are the stories that really keep you up at night. Let's talk about fat embolism.
This is a known complication of severe trauma, specifically fractures of long bones like the femur or major orthopedic procedures.
Why long bones?
Because the marrow inside your femur is yellow marrow. It's mostly fat. If you snap that bone, you not only break the bone, but you rupture the marrow and the thin-walled veins that run through it. Globules of liquid fat get sucked into the circulation.
And they travel to the lungs and brain and block small vessels. But the critique of just calling it a mechanical blockage is valid, right? Robbins goes deep on the biochemistry here.
You're absolutely right. It's a two-hit hypothesis. Hit one is mechanical. Yes, the fat globules physically block microvessels in the lung and brain, causing ischemia. But hit two is biochemical.
What's that?
The fat globules are broken down in the blood, releasing free fatty acids. These fatty acids are directly toxic to the endothelium. They cause chemical injury to the vessel lining all over the body.
So it causes a massive inflammatory storm.
A systemic inflammatory response. That's why you see the classic clinical triad of symptoms, which usually appears 1 to 3 days after the injury. First, hypoxia from lung injury. Second, neurological abnormalities like confusion or coma from brain injury. And third, the giveaway sign.
The petechial rash.
Tiny little red dots like pinpricks on the chest, neck, and shoulders. That's from thrombocytopenia and capillary bursting due to that widespread toxic endothelial injury. If you see a confused patient with a broken leg and that rash, you have to think fat embolism syndrome.
Then there's air embolism. The bends.
Yes, or caisson disease. This is all about the physics of gas solubility. At sea level, nitrogen is dissolved in your blood. If you are a deep sea diver, you are under high pressure, so more nitrogen dissolves into your blood and tissues.
It's like the carbonation in a soda bottle. Under pressure, the CO2 stays dissolved.
That is the perfect analogy. If you swim up to the surface too quickly, the ambient pressure drops suddenly. The nitrogen can no longer stay dissolved, and it comes out of solution, forming bubbles inside your blood vessels and tissues.
And those bubbles act just like solid clots.
They block vessels. If they form in your joints or muscles, they cause excruciating pain, the bends. If they form in your lungs, they cause respiratory distress, the chokes. If they cause chronic ischemic injury to bone.
That's caisson disease.
Right. Named after the pressurized caissons that workers used to build the foundations of the Brooklyn Bridge underwater. They would come up too fast and get crippling joint pain from ischemic necrosis of the bone caused by nitrogen bubbles.
And the last exotic one, amniotic fluid embolism.
This is a rare but catastrophic complication of labor and delivery. During a particularly difficult labor, there can be tears in the placenta and uterine veins, allowing amniotic fluid to enter the mother's circulation.
I always thought this was just fluid blocking a pipe. But Robbins makes it clear it's more like an anaphylactic reaction.
It is. It's a biochemical storm, not just a mechanical blockage. Amniotic fluid is full of fetal material skin cells called squames, lanugo hair, mucus, but it's also loaded with pro-inflammatory and pro-coagulant substances.
So when it hits the mother's blood,
It triggers a massive systemic activation of her immune and coagulation systems. You get intense pulmonary artery vasospasm leading to shock, and you almost always trigger full-blown DIC disseminated intravascular coagulation.
so the mother isn't just dealing with a blockage, she's dealing with a storm that causes her blood to clot and bleed at the same time.
The mortality rate is staggering, upwards of 80%. It's incredibly rare, but absolutely devastating when it happens.
Okay, we've talked about the blockages. Let's briefly touch on the result. Infarction, tissue death.
The end result of ischemia, ischemic necrosis.
There's a classic logic puzzle here that students often struggle with. Red infarcts versus white infarcts. It seems random at first, but it's actually purely anatomical.
It is. Let's break it down simply so you never forget it. White infarcts, or anemic infarcts, happen in solid organs that have a single end arterial blood supply. Think heart, kidney, spleen.
One road in, one road out. A cul-de-sac.
Exactly. If you block the only road in, the tissue downstream gets no blood. It dies, and over time it turns pale and scar-like because no new blood can get there. It's a white infarct.
And red infarcts.
These happen in two main situations, either in loose, spongy tissues like the lung, where blood can ooze into the dead area, or, more classically, in tissues that have a dual blood supply.
Give me an example of a dual supply.
The lung is a great one. It gets blood from the pulmonary arteries and the bronchial arteries. The liver has the hepatic artery and the portal vein. The small intestine has a rich network of anastomoses.
So if you block one of the roads,
The other road is still open. But here's the critical part. The backup road often isn't enough to keep the tissue alive, but it is enough to keep pumping blood into the area that is now dead and necrotic.
so the tissue dies, but gets saturated with blood from the secondary supply.
Precisely. It becomes a red, boggy, hemorrhagic mess. So the rule is, solid organ single supply equals white. Loose organ or dual supply equals red.
That clarifies it perfectly.
Yeah.
Okay, we have arrived at the final and perhaps most complex part of our deep dive. We've looked at local problems, a clot in a leg, a block in a lung. Now we look at what happens when the entire system crashes. Part 5, shock.
This is a topic that deserves a lot of respect. Shock is the final common pathway for a huge number of lethal events, from trauma to infection to heart attacks.
We need to define it very carefully.
Right.
It's not emotional shock. It's not just low blood pressure, although that's usually part of it.
No, the core definition is systemic hypoperfusion. It means the cardiovascular system as a whole is failing to deliver enough oxygen and nutrients to meet the metabolic demands of the cells. The entire body becomes ischemic.
We classify it into 3, maybe 4 main buckets. Let's run through the mechanics of each because the treatments are completely different. Bucket 1, cardiogenic shock.
The pump failure. This is straightforward. You have a massive heart attack, a life-threatening arrhythmia, or a valve ruptures. The tank, your blood volume, is full. The pipes are fine, but the engine is broken. It can't generate the pressure to push the blood forward.
Bucket 2, hypovolemic shock.
The empty tank. Again, simple mechanics. Massive hemorrhage from trauma, severe burns leading to plasma loss, or massive dehydration from something like cholera. Yes, a perfectly good pump, but there's nothing for it to pump.
And bucket 3, septic shock. This is the complex one.
This is a type of distributive shock. Here, the tank is full, And the pump is working, at least initially, but the pipes, the blood vessels have failed.
They've vasodilated.
Massively. Systemic vasodilation, the total diameter of your blood vessel container increases drastically.
And basic physics tells us that if you dramatically increase the volume of the container without adding more fluid, the pressure has to drop.
It bottoms out. It's like trying to pressurize a mansion's entire plumbing system with a standard garden hose. There's not enough fluid to fill the expanded space. But let's dive into the mechanism of septic shock, because Robbins goes really deep here. It all starts with the immune system.
PMPs.
Pathogen-associated molecular patterns. Things like lipopolysaccharide, or LPS, from the outer wall of gram-negative bacteria. Your immune cells, like macrophages, recognize these PMPs via toll-like receptors.
And they go nuclear.
They go absolutely nuclear. They unleash a cytokine storm, a flood of inflammatory mediators like tumor necrosis factor and interleukin-1.
And these cytokines are the signal that tells the blood vessels to relax.
They trigger a massive release of nitric oxide, which is a potent vasodilator. But that's not all. They also make the vessels leaky. They damage the endothelial lining, causing fluid to shift from the blood vessels into the tissues.
So it's a double whammy.
A double whammy for blood pressure.
Yeah.
The pipes get wider, and the fluid that's in them starts leaking out.
There is a really important clinical pearl here regarding the temperature of the patient's skin. In cardiogenic and hypovolemic shock, the patient is classically cool and clammy. Why is that?
It's all about compensation. The body senses the dangerously low blood pressure. It triggers a massive sympathetic response that clamps down on all the peripheral blood vessels, shunting what little blood flow there is to the vital core organs, the brain and the heart.
The skin gets no blood.
The skin gets sacrificed, it becomes cold and clammy, but in early septic shock,
It's the opposite.
the patient is often warm and flushed because the primary pathological mechanism is vasodilation. The pipes are wide open, the skin is flooded with warm blood, even though the vital organs are beginning to starve for oxygen. It's a crucial distinction to make in the ER.
Now, shock isn't a static state. It's a progression. It's a slide down a cliff. Robbins outlines the 3 stages, and this is where we really see the cellular tragedy unfold.
Stage 1 is the non-progressive or compensated stage. This is the body fighting back. You get tachycardia as the heart races to compensate. You get that peripheral vasoconstriction we talked about. The kidneys hold on to every drop of water. If you intervene here and fix the underlying problem, the patient usually survives.
But if you don't, you enter stage 2,
Mm-hmm.
the progressive stage. This is where the compensation starts to fail.
This is the critical turning point. This is where we switch metabolic pathways because oxygen isn't getting to the tissues. The cells can't perform aerobic respiration in the mitochondria, so they switch to their backup plan, anaerobic glycolysis.
Which is incredibly inefficient at making energy.
Terribly inefficient. You get a tiny amount of ATP. And more importantly, the byproduct is not CO2 and water. The byproduct is lactic acid.
So the patient develops a profound lactic acidosis.
The pH of the entire body begins to drop. And here's the lethal kicker. Acidosis blunts the vasomotor response.
What does that mean in simple terms?
It means the arterioles, which were trying their best to constrict to maintain blood pressure, stop listening to the signals from the brain and the nervous system. The acid essentially paralyzes them. They give up and dilate.
So just when you need that pressure the most, the vessels fail.
The vessels fail. Blood pools in the periphery. Cardiac output drops even further. It is a vicious, accelerating downward spiral.
And finally, stage 3, irreversible shock.
This is the point of no return. Even if you could magically fix the blood pressure and oxygen delivery right now, the patient would still die.
Why? What has happened at the cellular level?
The cells themselves are broken beyond repair. Without ATP, the ion pumps in their membranes have failed, and critically, the lysosomes, the little bags of digestive enzymes inside the cell rupture,
They release their contents.
they release digestive enzymes into the cell's own cytoplasm. The cells literally begin to digest themselves from the inside out. At the same time, the gut barrier fails. Bacteria from your own intestines flood into your bloodstream, adding a huge septic load to the fire. It is multi-organ dysfunction syndrome, or MODS. Game over.
It's terrifying. And in the middle of this septic chaos, we often see a condition we mentioned earlier, DIC.
Disseminated intravascular coagulation. It is the ultimate cruel paradox of the dying system. The cytokine storm and endothelial injury are so intense that they activate the clotting cascade everywhere all at once in the microcirculation.
You get millions of tiny clots blocking up all the capillaries in every organ.
Which of course worsens the oxygen delivery even more. But because you're frantically making millions of clots all over the body, you use up all your platelets and clotting factors. You literally run out of the building blocks for clots.
This is the consumptive coagulopathy.
Exactly. So now, with no platelets or factors left, the patient starts bleeding spontaneously. They bleed from their IV line sites, from their gums, from their gut. They are clotting to death and bleeding to death at the exact same time. It is one of the most difficult and terrifying conditions to manage in all of medicine.
It really brings home the central theme of this entire deep dive: balance. The very systems that are designed to keep us alive, clotting, inflammation, vascular tone, become lethal when they lose their regulation.
Pathology is just physiology gone rogue. That's all it is. The body tries to save you by clotting a bleed or dilating vessels to bring immune cells to an infection. But the response is so overwhelming and unregulated that it destroys the logistics of the whole organism.
So let's synthesize. We've covered a tremendous amount of ground.
We started with the physics of fluids, that tug of war between the hydrostatic push and the oncotic pull. When that fails, we get edema.
We moved on to the solid state, thrombosis, all governed by the elegant framework of Virchow's triad: endothelial injury, stasis, and hypercoagulability.
We then watch that clot travel as an embolus, whether it's a thrombus or fat or air or amniotic fluid, turning a local problem into a distant catastrophe like a pulmonary embolism.
And finally, we witnessed the systemic collapse, shock, the predictable slide from compensation to anaerobic metabolism, lactic acidosis, and irreversible cell death.
It's a heavy topic, but understanding these logistical failures is the absolute core of critical care medicine. It's the difference between life and death in the ICU.
I want to leave our listener with a final thought. We tend to think of disease as something that causes pain or symptoms, but so many of these hemodynamic disorders are profoundly silent. A DVT is asymptomatic in 50% of people. Atherosclerosis builds for decades without a single whisper.
It really emphasizes why we study mechanism. You can't just wait for the event to happen. You have to understand Virchow's triad to predict who is at risk. You have to understand the physiology of shock to catch it in that warm and flushed phase before it becomes cool and clammy and irreversible. We study pathology to learn how to see the invisible threats.
That's the mission. All right, your homework for this deep dive. Open Robbins. Find figure 4.1, the diagram of Starling forces and edema. Find a color photo of the nutmeg liver and the lines of Zahn. And really read the section on the stages of shock. It's dense, but it's the story of how life ends and how we can sometimes stop it.
The visuals will anchor these concepts for you. It makes a huge difference.
Thanks for diving deep with us. Keep the flow laminar, keep the pressure regulated, and we'll see you in the next deep dive.
Take your